Dear students, Assalamu alaikum. Today the topic of our presentation is organization of the photosynthetic apparatus. Photosynthetic apparatus basically that is the chloroplast present in the green plants, which is the site of photosynthesis. It contains its own DNA, RNA and ribosomes. Now, as we all know, green plants are the only biological source which can capture the sunlight and can convert it into the chemical energy which is required for the normal growth and functioning of the green plants as well as for the rest of the life on this universe. Now, the most active photosynthetic tissue in higher plants are the mesophils of the leaves. Mesophyll cells contain chloroplast, which contain the specialized light absorbing green pigments, the chlorophyll molecules. Now, chloroplast, this is the structure of chloroplast, a double membrane organelle. Now thalakide are present inside the chloroplast molecule, that is the stacking of the membranes and looking just like a stacked like coins. Thalakide is the region where the light reaction of photosynthesis occurring. Now thalakide stacking when occurs, so the region where the overlapping region of the thalakide membrane is termed as the thalakide lumen, whereas surrounding the thalakide membrane there is a region of the dark reaction which is termed as the stroma. These are the stroma regions, whereas stroma and thalakide are connected with the stroma lamella. Now, the stacking of the thalakide membrane is termed as gram membrane. Now, we can say thalakide contain integral membrane proteins. Now, as containing the integral membrane protein, definitely it must having the amino acids, and in this picture we can see the overlapping region of the thalakide which is termed as the thalakide lumen. When in this diagram there is a small piece has been taken and this is the big picture of that small piece. Thalakide membrane contain integral membrane proteins which composed of amino acid and amino acid having amino group as well as carboxyl group. The amino group of the integral membrane proteins that are toward the stroma region, whereas the carboxyl functional group will be toward the thalakide lumen. Next is photosynthetic pigment absorb light that power the process of photosynthesis. Now, as the sunlight is first absorbed by the pigments, green pigments in the plant, all the pigments active in photosynthesis are found in the chloroplast. So मतलब ये कि different photosynthetic organisms have different type of the photosynthetic pigments as shown in this graph 7.7. The first line of the black color, that is bacteriochlorophyll, the chlorophyll molecule present in the photosynthetic bacteria, and it absorbs the light coming from the sun ranging from almost below 100 till the 800 nanometer. Whereas the second line, that is representing the chlorophyll A content present in the green plants, and this second line is of the green color, that is chlorophyll A molecule absorbing light from 400 nanometer up to the 700 nanometer. The third one line is of the chlorophyll B molecule that is also present in the green plants, which has been shown with the red color, and it absorbs light from 400 nanometer and less than 700 nanometer. The fourth one line, which is of the violet color, absorb light from the 400 nanometer up to the 600 nanometer, and that fourth line is of the phycoerythrobilin, whereas the fifth line is of the light blue color, that is carotenoid content present in the green plants, which absorb light ranging from 400 nanometer up to the 500 nanometer. The next is chlorophyll molecules are present in the photosynthetic organisms, whereas in the green plants chlorophyll A and chlorophyll B are present abundantly, whereas the chlorophyll C and D are present in the protists and cyanobacteria. Now, the bacteriochlorophyll molecule is almost similar to the chlorophyll A molecule of the higher plants. All the chlorophyll have a complex ring structure that is chemically related to the porphyrin-like group found in the hemoglobin and the cytochromes, whereas a long hydrocarbon tail is attached to the porphyrin like ring structure in the chlorophyll molecule. The tail purpose is to anchor the chlorophyll molecule to a hydrophobic environment. Now the ring structure having loosely bounded electron which are involved in the redox reactions. As we know, photosynthesis is a redox reaction, mean to say a continuous process of oxidation reduction reactions. So that is because of the loosely bounded electron in the ring structure of the chlorophyll molecule. The different type of carotenoids found in photosynthetic organisms are all linear molecule with multiple conjugated double bond that has been shown in the figure 7.6b. Because the chlorophyll molecule is not having the multiple conjugated double bond, only carotenoid content having the multiple conjugated double bond. Carotenoid content, we can say, that is absent in the mutant. In the cultured plant, carotenoid content is absent, and carotenoid content as integral constituents of the thylakoid membrane are associated with the antenna system as well as with the reaction center. The light absorbed by the carotenoid content is transferred to the chlorophyll molecule because that is not connected directly to the reaction center. That's why we can say carotenoid contents can be termed as accessory light harvesting pigment, because it can harvest the sunlight, can capture the sunlight, and can shift it to the chlorophyll molecule, and that chlorophyll molecule then shifting that captured energy being transferred from the carotenoid to the reaction centers. So mean to say, carotenoid contents are not directly in contact to the reaction site. Next is as the diagram indicating the chlorophyll molecule, chlorophyll A molecule. This is a porphyrin like ring structure, whereas that is a long hydrocarbon tail attached for entering into the hydrophobic environment, whereas the carotenoid content, as we discussed, having a multiple conjugated double bond. Next is the absorption spectrum of the chlorophyll A molecule indicates approximately the portion of the solar output that is utilized by the plants. Now in this diagram, it has been shown the absorption spectrum. Now the sunlight coming ranging from almost 0 to 2000 nanometer, whereas the fraction of light which is reaching to the earth surfaces, that is ranging from less than 400 nanometer and reaching up to the 2000 nanometer, and this has been indicated by the red color line, whereas the green line, that is the absorption spectrum, the fraction of light which is going to be absorbed by the green plants ranging from 400 nanometer almost reaching up to the 700 nanometer, and this that has been indicated by the green line. Now what is the absorption spectrum? So absorption spectrum is the amount of light which can be absorbed by the green plants. So mean to say, it display the amount of light energy taken up or absorbed by a molecule, and that will be the function of its wavelength. Next is when molecule absorb or emit light, they change their electronic state. Now this is as we know, the light coming from the sun will be either in the form of wave or in the form of particles. If that is in the form of particles, that particle will be termed as the photon. Photon carrying energy from the sun, that will be represented by this equation. So the light once striking the chlorophyll molecule can excite the chlorophyll molecule, and this is the representation of the excited chlorophyll molecule. This one is the ground position chlorophyll molecule, whereas this one is the excited position of the chlorophyll molecule. Whereas once the molecule excited from the ground position, the distribution of electron in the excited molecule will be different from that of the ground position. So mainly the visible spectrum of the sunlight carrying the 7 colors, among which the blue and the red light are considered effective for the process of photosynthesis, but as far as the blue is considered more energetic as compared to the red light. So the blue light can excite the chlorophyll molecule up to a maximum range as compared to the red light. Now once the chlorophyll molecule reach to the excited position, at that particular condition, that molecule are considered unstable, and that molecule want to come back to its ground position. In that particular case, when the excited molecule coming back to the ground position, it will emit its energy, and that is the emission of that captured energy required some several mechanisms. For this purpose, there are several mechanisms. Mainly, the four mechanisms are important. The first one is excited chlorophyll molecule. When coming back to its ground position, it will re-emit a photon that is not like the photon coming from the sunlight. That is somehow a secondary photon of carrying less energy, and this phenomena is known as fluorescence, when the excited chlorophyll molecule emitting that energy in the form of secondary photon. The second mechanism is the excited chlorophyll molecule can return to its ground position by directly converting its captured energy in the form of heat, and there will be no emission of a photon, that is just dissipation in the form of heat energy. The third one is chlorophyll molecule can transfer its energy to another molecule, and that is the third mechanism. The fourth one is photochemistry, when the energy being captured by the chlorophyll molecule, and when that excited chlorophyll molecule reaching back to its ground position, the energy will be utilized for the photochemical reactions, mean to say the chemical reactions. So this phenomena is important in the process of photosynthesis because the splitting of water molecule is required to release the electrons and protons from the water molecule, and that splitting of water molecule, which is termed as photolysis, is only possible with the help of captured energy coming from the sun. So this photochemistry is important for splitting up the water molecule, the fourth mechanism. Now in this diagram, this has been shown. The blue color, the blue range, has excited the chlorophyll molecule up to a higher position as compared to the red light. So coming back to the ground position from this position, it will take time, whereas from this region, when the excited chlorophyll molecule, which is termed as the lowest excited state, coming back to the ground position, will be in the form of fluorescence, yeah, we can say the secondary photon, whereas from the higher excited state, because of the blue light, when coming back to the ground position, it will lose energy in the form of heat, whereas will also emit the secondary photons. So this diagram indicating that blue light can excite the chlorophyll molecule up to a higher position as compared to the red light. Now the next is action and absorption spectrum. What is action spectrum and what is absorption spectrum? So action spectra relate light absorption to photosynthetic activity. Now action spectrum has been central to the development of our current understanding of photosynthesis. An absorption spectrum display the amount of light, as already discussed in the previous slides. Absorption spectrum display the amount of light energy absorbed by a molecule or substance, and that is the function of its wavelength, whereas the action spectrum portray the magnitude of response of biological system to the light, and this is also the function of its wavelength. Now the action spectrum can be measured, how much the amount of energy is going to be utilized in the process of photosynthesis, that has been measured by the amount of oxygen evolved during the process of photosynthesis, and once the amount of oxygen evolved under the consequences of the process of photosynthesis, so that amount is directly proportional to the action spectrum, and we can say that if the maximum amount of oxygen is going to be evolved, so mean to say the maximum amount of light energy captured is going to be utilized in the process of photosynthesis. In this diagram indicating absorption spectrum, whereas the one is the action spectrum. So absorption spectrum basically can be larger, whereas action spectrum almost will be lesser than the absorption spectrum, because the whole amount of energy once absorbed by the green plants will not carry in the process of photosynthesis. Lesser than the absorption portion, small amount of light can carry the process of photosynthesis, because the absorption spectrum amount of energy can be used in some other biological processes also. Now this is a simple experiment has been performed by the Engelmann in the late 80s, indicating the action spectrum. For this purpose he used the prism. He dispersed the sunlight into a rainbow, which is composing into 7 colors, and that 7 colors has been used for the purpose of photosynthesis in the aquatic algal filamentous. That aquatic algal filamentous has been placed in front of the dispersed light coming from the sun, whereas on the other side he used oxygen seeking bacteria aggregation. Yeah, we can say the congregation of these oxygen seeking bacteria will be maximum in the regions where the process of photosynthesis can occur, because oxygen is evolving as a byproduct in the process of photosynthesis. So once light falling on the aquatic algal filamentous can carry the process of photosynthesis, and that emitting the oxygen, and that bacteria seeking oxygen seeking bacteria will be congregated in the region where the maximum photosynthesis rate occurs. So the blue and the red spectrum is considered effective in the process of photosynthesis and can emitting the maximum amount of oxygen. So we can say this is the action spectrum required for the normal function of the